Hello everyone, welcome back to Biome, Biology, Dilse. Today we will discuss about the double fertilization in angiosperm. So the detailed process of the double fertilization and the triple fusion already we have discussed in the last video. So if you missed that video, please go back into the playlist of the class 12th and there also you can find out the video of the double fertilization and the triple fusion. Now here in this video particularly we'll discuss about how the endosperm are formed. But before directly going into the video, there is just a message from my side. Please subscribe the Biome channel. आप लोग देखते हैं वीडियो बट आप लोग सब्सक्राइब नहीं करते हैं. Actually, subscription gives us lots of energy. Okay, it motivates us to make a new video time to time. And please subscribe the channel and also please hit the bell icon also. And if you hit the bell icon, so you will also get the notification about the new videos. And बेल आइकन में जो घंटी वाला बेल आइकन होगा ऊपर वाला बेल आइकन है उसको तुम लोग जरूर प्रेस कर लेना और वीडियो के बीच में जो आई बटन होगा उस आई बटन को भी प्रेस करके तुम लोग जो अदर वीडियो है उसको भी देख सकते हो. So please once again do not forget to subscribe the Biome channel and please share the content of this Biome channel with your peer groups also, with your friends also. Okay, so please help us to grow. So now without wasting of time, directly we'll come into our today's topic, that is the double fertilization in plant, especially in the flower. So once we come to the concept of the double fertilization, so first thing it comes in our mind, what is this double fertilization? What is this? So this double fertilization generally, and where we'll get this double fertilization? Generally, this double fertilization generally, definitely where we'll get? Definitely we are getting in plants, correct? But this double fertilization, what is this? Simple, the fertilization we means that one, the fusion of male gametes and female gamete. That is the simple fundamental idea behind the fertilization. So that's why here the double fertilization, but here it's a double. So the double fertilization is a complex process which involves the fusion of one of the female gametophyte with two male nucleogametes. So that we'll have to discuss about these things only. So how it is possible? So before directly going into this double fertilization events, so first we'll discuss about who actually discovered this entire process of this double fertilization, and afterwards again will come the detailed process of the double fertilization. Means in summarize from, if we come to the double fertilization, it's a very much complex process where the male nucleus, okay, it is going to fuse with the female nucleus. So that is the detailed process of the double fertilization. So this double fertilization process, it's actually first discovered by the Navaschin in the year of 1898. Okay, in the Liliaceae plant, generally the name of the plant is the Lilium mortagon and the Fritillaria tenila. Okay, so this process, मतलब ये जो double fertilization process है, first Navaschin discover किए थे from the Liliaceae plants, and the scientific name of the Liliaceae plant was the Lilium mortagon and the Fritillaria tenila, and this plants looks like this, and this is the actually the scientist Navaschin, and this is the our Lilium mortalum, and here our Fritillaria tenila. So these are the actually here Lilium mortagon and the Fritillaria tenila, and this is the picture of the Navaschin. So the Navaschin first discovered, this biologist first discovered the process of double fertilization. Double fertilization first discovered by the Navaschin. Later this events of the double fertilization, it's confirmed by Gargnat. Okay, later it's confirmed by Gargnat. So first कौन discover किया था? Navaschin in the year of 1898, and later it is get confirmed by scientist Gargnat. तो इतना मतलब क्या है, डबल फर्टिलाइजेशन कौन डिस्कवर किया, नवास्टीन। कौन सा प्लांट से डिस्कवर किया, वेरी इजी, those plants belongs to Liliaceae family. So first plant was the Lilium martagon and the second plant was Fritillaria tenila. From these plants, the scientist Navastin first discovered the events of double fertilization, and later the events of double fertilization is confirmed by the scientist Grognut. So let's move into the detailed process of the double fertilization. So comes to the process of the double fertilization. So generally the flower is the most fascinating character of the angiosperm, correct? The flowers are the most fascinating reproductive characteristics features of the angiosperm, and especially those are the terrestrial plants. They are showing a varieties of the flowers also, and their adaptation way of double fertilizations also varies. That means they have a much more diversity in the term of double fertilization. So now let's look into this flowchart. So here just I made a simplified flowchart for you. Okay, so whatever the process we will discuss, so that is the extract of that particular topic. So यहाँ पे हम लोग अब जानने का कोशिश करेंगे तो कि इस टॉपिक के अंदर में कैसे हम लोग इस डबल फर्टिलाइजेशन प्रोसेस को अच्छे तरीके से समझ पाएंगे. So let's move into the picture. So this is the actually the picture of the double fertilization. So once we come to the picture of the double fertilization, so what we can get? So especially come to the topic over here today. So the topic first things, this is we know already a germinating pollen grain, and this is nothing but the pollen tube. This is nothing but pollen tube, and this is nothing but the style, and this portion is nothing but our ovule, करेक्ट. तो यहाँ पे डबल फर्टिलाइजेशन कहाँ पे होगा, तो डबल फर्टिलाइजेशन का जो डिटेल इवेंट है दैट इज एक्चुअली टुक्स प्लेस इनसाइड द ओव्यूल ओनली. तो कैसे हम लोग समझेंगे, प्लीज लुक इनटू दिस पिक्चर वेरी नाइसली. In this base portion, we are getting two male nuclei. So here we are getting how many? Two male nuclei we are getting. यहाँ पे हम लोग को दो मेल न्यूक्लियस मिलता है. इसका मतलब क्या है, सिंपल फंडा है. हम लोग को पोलन ट्यूब के अंदर में हम लोग को कितना मिलेगा, हम लोग को दो मेल न्यूक्लियाई मिलेगा, करेक्ट, दो मेल न्यूक्लियाई मिलेगा. तो अब डिटेल इस प्रोसेस में आएंगे, हम लोग इस पिक्चर में आएंगे. तो यहाँ पे देखो क्या है, दो मेल न्यूक्लियाई है, करेक्ट. तो ये सब क्या है, हम लोग ओव्यूल के अंदर में, चलो फटाफट इसको हम लोग नाम दे देते हैं. सो व्हाट इज दिस, दिस ऑल आर, वी नो दिस ऑल आर antipodal cells. Okay, these are the antipodal cells. So what is this, these two are the nothing but the synergids. Okay, these are the nothing but the synergids. And what is this, these are the nothing but the egg. And what is this two, these two are nothing but the polar nuclei. These are the nothing but the polar nuclei. अब यहाँ पे हम लोग सारा चीज को हम लोग देखेंगे कैसे इस जगह में फर्टिलाइजेशन होता है, and what is the detailed events of the fertilization. So for that one, let me erase the entire portion, then we will again proceed into the main topic. So now look into here, दो मेल न्यूक्लियस के अंदर में एक मेल न्यूक्लियस इस एक के साथ फ्यूज होगा, एक के साथ फ्यूज होगा. What is the ploidy of this egg? This ploidy of the egg is haploid. अब इस haploid and यहाँ पे मेल न्यूक्लियस का ploidy क्या है, this is also haploid. अब ये दो मेल न्यूक्लियस फ्यूज होगा, तो इस फ्यूजन को हम लोग क्या बोलेंगे, डेफिनेटली दिस इज कॉल्ड एज फर्टिलाइजेशन. Definitely this is called as fertilization, and we can also call this one as syngamy, correct. So now due to this fertilization, what will happen, there will be formation of zygote. There will be formation of diploid zygote. इसका मतलब क्या है? इस एरिया में मेल न्यूक्लियस जब फीमेल न्यूक्लियस के साथ फ्यूज हुआ तो तब जाके क्या बना? जाइगोट बना। और ये जाइगोट का प्लॉइडी क्या होगा? डिप्लॉइड। And that's why you are getting over here a diploid zygote. And remaining other male nuclei, it's going to fuse with the polar nuclei. अब यहाँ पे पोलर न्यूक्लियर में हम लोग प्लॉइडी लिखेंगे। यहाँ पे क्या है? दिस इज वन हैप्लॉइड, दिस वन आल्सो हैप्लॉइड एंड दिस प्लॉइडी ऑफ दिस मेल न्यूक्लियर, दिस इज आल्सो हैप्लॉइड। तो इस जगह में क्या होगा? 3 हैप्लॉइड न्यूक्लियस का फ्यूजन हो रहा है इस एरिया में। Correct? So that's why here, as it's fusing the 3 male nuclei, total 3 nuclei, that's why it's called as triploid. That means it's a 3N. And once they will fuse, so there will be formation of endosperm. इसके मतलब क्या है? एक remaining male nucleus दो polar nuclei के साथ fuse होके किस चीज का formation होगा? Endosperm. Correct? So इसका मतलब क्या है? इसमें का मतलब क्या है? सिनगैमी के जरिए किस चीज का फॉर्मेशन होगा? जाइगोट का फॉर्मेशन। अब इस जगह में क्या होगा? फर्दर जाइगोट का क्या होगा? फर्दर जाइगोट का डेवलपमेंट होगा। ओके, फर्दर जाइगोट का क्या होगा? डेवलपमेंट होगा। अब जो यहाँ पे एंडोस्पर्म बना, ये कैसे एंडोस्पर्म बनता है? तो यहाँ पे फ्यूजन के कारण में, okay, the fusions of the two male gametes, or sorry, over here one male gamete, and over here also with the male gametes the polar nuclei also fused, and due to that one there will be formation of PEN. So what do you mean by the PEN? The PEN means it's a nothing but it's a peri endosperm nucleus. क्या हो रहा है यहाँ पे? What is the full form of PEN? The full form of PEN is nothing but peri endosperm nucleus. Later क्या होगा? Later this PEN later it's located inside, okay, or it will be converted into PEC. Later क्या होगा? PEN किस में convert हो जाएगा? PEC. So generally what happens? The PEN actually located within the primary endosperm cell. So what is this PEC? PEC means nothing but it's a primary endosperm cell. So the pen, it will be get converted into PEC. Pen कैसे तैयार हो रहा है? The one male nuclei it's going to fuse with the polar nuclei, and due to that one there will be formation of pen, means peri-endosperm nucleus, and again the peri-endosperm nucleus it's going to convert it into PEC, that means the peri-endosperm cell, and this peri-endosperm cell again it will be get converted into endosperm. Now question is that in this plant what is the role of this endosperm? Now इसके लिए हम लोग यहाँ पे क्या देखे हैं? ये जो जाइगोट है, जाइगोट का डेवलपमेंट होगा। तो जाइगोट का डेवलपमेंट के लिए जाइगोट को क्या चाहिए? कुछ खाना चाहिए, कुछ न्यूट्रिशन चाहिए। इस न्यूट्रिशन कौन प्रोवाइड करेगा? ये न्यूट्रिशन प्रोवाइड करेगा एंडोस्पर्म। So that's why what happens? These endosperms are play very crucial role, okay, for the development of the zygote, okay, into embryo. Correct? So what is the role of the endosperm? The endosperm actually play very crucial role, or it gives nutrition to the zygote, okay, and after using the nutrient from the endosperm, the zygote will be get converted into embryo. So next we'll learn what will be the fate of the endosperm and what will be the fate of the zygote. So what is the fate of the zygote? So definitely what we came to know that one, the zygote it's actually diploid, and later this zygote is going to develop into embryo, and this development of the zygote into embryo, so this is called as embryo embryogeny. So why it's a embryogeny? The embryo means definitely it's a actually it's indicate the embryo, and this geny words it came from generator. So the generator means, so definitely here the embryo is forming. So that's why this is called as embryogeny. So what is the embryogeny? The development of the zygote into an embryo. It's called as an embryogeny. Now, what are the process will be there in the, or what are the process involved in the zygotic development? That means what are the process involved by which the zygote will be converted into embryo, or also we can write what are the process of the embryo, what are the process of the embryogeny. So let's learn the process of the embryogeny with the help of a picture. So this is the picture. So far what we have discussed, we have discussed two three concepts. The first concept over here, we already know the primary endosperm nucleus will form. Primary endosperm nucleus is going to form into PEC, and this PEC or primary endosperm cell later it will be converted into endosperm. हम लोग को इतना तो ऑलरेडी पता ही है. And what endosperm का काम क्या है? Endosperm will provide nutrition to growing to zygote, एंड जाइगोट इस न्यूट्रिशन को इस्तेमाल करने के बाद जाइगोट किसमें कन्वर्ट हो जाएगा? धीरे धीरे एम्ब्रायो में कन्वर्ट हो जाएगा. तो ये जो इंटर लार्ज पोर्शन है इसको हम लोग एक छोटा सा पोर्शन में एज्यूम करते हैं. तो धीरे धीरे क्या हो जाएगा? ये जो जाइगोट है धीरे धीरे एम्ब्रायो में कन्वर्ट होगा, एंड दैट प्रोसेस इज एक्चुअली कॉल्ड एज एम्ब्रायोजेनी. So first, as you are getting over here, the nucleus of the zygote, it will be get divided, okay, by the different process of the cell divisions, and as a result we will get some intermediate stages we will get. So first we will get that one, the zygote will be get converted into embryonal cell. So what we will get? So the zygote will get converted into, zygote will be converted into embryonal cell. Embryonal cell to over here, as we are getting here, the two it will be there, two nucleus is formed. From the two nucleus there will be again four nucleus will form. From the four nucleus there will be again eight nucleus will form. So like this, so there will be embryonal cell will form. From the embryonal cell, from there will be formation of tetrad stages, tetrad stage. From the tetrad stage there will formation of octant stage. So these are the intermediate stages, and from the octant means it's nothing but 8. So from the octant state there will be formation of one globular embryo. So as you are getting this picture, this lower portion is nothing but the circular like, so that's why this is called as globular embryo. So there will be, from the octant stage there will be formation of the globular embryo. So before the globular embryo, I have written already the octad and the tetrad. Here actually it's not mentioned. You will learn this one in the higher classes. What is the detailed process of this development? But for our standards, we can learn up to this much. From again the embryonal or your globular embryo will be get converted into a next stage, that is the heart-shaped embryo. Why? Because if you look into this portion, these portions, if you look, they looks like a heart. So that's why this is called as a heart-shaped embryo. So this is called as heart-shaped embryo. So later this heart-shaped embryo, we are getting that one, they will be converted into a mature embryo. They will be get converted into an mature embryo. So these are the detailed process of the embryonic development. And जो mature embryo होगा, mature embryo के अंदर में क्या होगा? Definitely the radical होगा. That radical, that means this is the nothing but the few going to be converted into, these are the future root system. We already know this one. These are the future root system, and the plumule, that means this is nothing but the future shoot system, and this cotyledon we already know, the reserve reserve food material. It reserved food materials. So these are the actually parts of the mature embryo. So one more, this entire zygote, it's going to convert it into first the embryonal cell to the tetrad stage to the octant stage to the globular embryo to the heart-shaped embryo, and heart-shaped embryo later again it will be converted into mature embryo, where in the case of dicot, in the case of dicot. But in the case of the monocot, this development up to some stages it's same, but in the later life of the development there will be little bit difference that we will learn in the coming lecture. So today up to this much, and if you like this video, so do not forget to subscribe the channel, okay, and please hit the once again bell icon, and please, if you want to see a new videos, please you can write in the comment box also, in especially in which topic I should make the video. I'll try to make the video on those topics, on those demanded topics. So till then, thank you. So thank you for watching our channel, that is biome only, and once request is there, please subscribe the biome channel and please hit the bell icon and subscribe it as soon as possible, and please spread the videos of this channel along with your friends also. So मिलते हैं अगले वीडियो के साथ, चलो, goodbye.